right. So, okay. Yeah, let me share my screen for caps or just for the capstone questions and stuff like that. Though it looks like we really don't have too much. What is this document? Oh, that's the wrong one. I know, oops, I'm sharing the wrong thing, aren't I? Can you guys see capstone questions? No.
Yeah, I can see it. Oh,
Oh, it
now it
was,
went away.
sorry. I thought that's weird. It usually shows a little green thing around it. There it goes. Where did you guys go? There you are. Okay, so cool. I know Gina had like wanted to ask a question and stuff like that. So I think that's the only thing that's on here that someone put down. So yeah, do you guys? Otherwise, I can talk a little bit about Git and stuff like that. But really, it's up to you. Whatever you guys want to talk about. If you guys have any specific questions, we can definitely go for that. So since Gina, you're here and you put something down, would you like to, ask your stuff first?
Okay, so I'm trying to decide how to put this and also have it be useful to other folks on the call. And I will be talking to Jeff at 2, so I guess I can get into more deep dive there. But let me share my screen. That may be, oh,
Yeah, I
I
think I see
cannot
it.
share screen while other participant is sharing.
There
Sorry
you
about
go.
that. Okay. All right. I'm just going to share my desktop and, whoops, sorry. I have to move this out of the way. Okay. So, as a quick overview for everybody, this is a competition on the Driven Data webpage. So I'm going to scroll up here. Driven Data, so they have a lot of competitions that are around some projects that have a social element to them, or a positive social good. This project, and Driven Data, I don't know if they're a nonprofit. They do work with a lot of nonprofits. This project, though, is hosted by Alt Labs, and what it is essentially machine learning models to try to identify where certain DNA molecules called plasmids are coming from, which labs in the US or in the world they're coming from. And so without getting into too much depth, the reason why you want to do this is because in a way it's almost like fingerprinting, because as you can imagine, while most of the producers out there are good actors, there can be some very bad actors. And also there's a notion of genetic plagiarism. I know it sounds crazy, but people may have patents on certain sequences, and are other labs basically using those sequences without the proper attribution or licensing, let's say. Okay, so the key thing with this, so there are most of the features of this data set are actually binary. So all of them except for one are binary. The one that is not is the sequence string. And so DNA plasmids are circular molecules that are generally put into some kind of a bacterium. This is a very common thing that's done in biotechnology. A lot of, for example, ingredients, medicines, things like this are often produced in this way. So when it says genetic engineering attribution challenge, we're not talking GMOs here. That's kind of a whole separate ballgame, just to put people's minds at ease. Also, what it means is that although this is hardly a simple problem, plasmids are pretty simple molecules, and so there's not the kind of complexity in them that you would find in like chromosomes, whether it's humans or fruit flies or mice or whatever. Speaking of which, one of the binary, a group of the binary, the dummy variables have to do with what species the plasmids are ultimately used in after they're cloned. Okay, so basically all we need to know here at the moment is almost all of these, except for one, as I mentioned, are binary, and the sequence is the only thing that is not. This is an extremely short sequence. Most plasmids are in the range of, I'd say on average, about 4,000 individual letters. So you've heard of DNA base pairs. Each individual letter corresponds to a base pair. And although DNA is double-stranded for the purposes of sequencing data, you read it in one direction, and the opposite strand of the DNA is actually complementary. So you can essentially, it's the same forwards as it is backwards. Okay. Now, the challenge is to predict which lab a sequence comes from. And so there are over 1,300 labs in this data set. So off the top of your, like initially you think, oh my God, there's 60,000 sequences in the database and over 1,300 labs. So to make this simpler, what Driven Data did was they said, use some starter code that we give you to assign a probability to whether or not each lab of the 1,300 actually produce this, but then take the top 10. For each sequence, the algorithm that they give you will, essentially turn it into probabilities and grab the top 10 probabilities. And then how the submission works is that if the actual lab corresponds to one of your top 10 for each sequence, then that you'll get a correct marking. So in essence, you've gone from trying to do a 1 in 1,300 to a 1 in 130 assignment. And what's interesting is they actually give you starter code. Okay, so pardon me for rolling up here. So they give you some starter code, and for example, here's a distribution of the sequence length. There are a lot of little tiny ones, but you could see here histogram, most of them are in here. There are some big ones. Okay, now they also do a histogram of the binary features, so you can see what's very commonly found in plasmids, and then things that are a little less commonly found. Test set. So as I said, these are the sequences, and normally, for those of you who've had any biology, you'll know there are 4 base pairs, A, T, G, and C. You may also see an N in here, and they put N into this data set because N can represent any one of those 4. So that is the fundamental problem. And then what they do is they get you going with some starter code, which is really helpful. It would have taken me a very long time. I could have done it, but just spending all this time on this is kind of crazy. So then I got worried. I'm like, okay, they have this blog posting where they kind of set up a lot of this stuff to begin with for you. And then they run a random forest model. And the predictive value is a lot better than what we could come up with just guessing, of course, as I mentioned. But it's far from great. And so initially I was like, oh my God, well, if my skills were such that I could only get this far, then obviously I wouldn't be able to do this as a capstone because all the work is already done. And not all the work, but a very initial starting point. But then, of course, you realize, oh, there's tons more work that can be done. And so, so I have a number of questions I won't go into here. And in the interest of time for my capstone, I'm just going to have to accept that my accuracies aren't going to be that high. I did achieve a little bit of better accuracy than what they had here. Simply, so they have feature engineering, and just briefly what they did was they said, let's just do, what do you call, permutations of 4-letter sequences. So you basically say, okay, you come up with all the permutations for 5 base pairs, and then you say, for each sequence, how often does each of these combinations appear? as well as how often do these binary dummy variables appear. Okay, then they have all this stuff, and then they run the random forest, and they don't do any, grid search CV or anything like that. And they come up, I mean, this is just close to 20% guessing, or the model comes up 20% correct with 1,300 labs. That is pretty impressive. It doesn't sound that great, but that's pretty darn good. Now, the top 10 scorer accuracy gets closer to 40%. So what I did, and then they do, so that's their accuracy, and then they do predictions. What I did was I took a variant of their approach, and then I was a bio major many, many, many years ago. And I don't remember anything other than the fundamental concepts about how certain things work and are done in plasmids, and that there are sequences in data in plasmids, DNA sequences that certain molecular tools recognize. So I created my own list. It's a very short list because of time. And so I came up with, well, first thing I did was 3-letter sequences rather than 4. And when I ran my model, not too bad, almost what they had, just doing, and there's a reason why I picked 3, and I won't go into that. Then I had selected sequences, and let's see, yeah, where I basically just set up a list of different things that have to do with some of these tools I mentioned. And it didn't do much better, and I was kind of bummed. And then actually when I combined their stuff and my stuff, it did even worse. So that was random forest. And this is my, I wanted to not spend too much time introducing this. Maybe it'll be helpful to somebody. But the key thing with DNA, there's a lot of complexity, one of which is where do you start reading? Because obviously that'll affect what you do. For every 3 letters, that can correspond to an amino acid when building a protein. So the reading frame is really important. In a way, kind of a spoiler alert, someone in the forums found a paper written by Alt Labs people. They apparently used a recurrent neural network. And part of me, I don't want to just jump right to that, because not that it's cheating. Like Jeff told me, in theory, you could even see if you can recreate someone else's work. And as long as you've done something other than just exactly copy their code and everything, that should be okay for capstone. But I'm struggling here. Like, we've gone through all these different modeling approaches in the curriculum, and yet I find myself going, geez, what would SVM do? Is that a good approach to use? I mean, it seems pretty intuitively, it seems like it has to be something like a neural network. But I guess I'm kind of grasping to try to figure out what would be the best, or what can I expect from different modeling approaches? For example, my intuition says random forest is not great for this, despite being way better than just randomly picking stuff. So thank you for indulging me in my rather lengthy description. And I really welcome anyone's thoughts on this. And I'll stop share unless, Victor, you think I should keep, I mean, I guess I could keep it open on the
Keep it up,
main,
I think.
yeah, competition page. Yeah.
No, thanks, Gina. It's really good to hopefully, obviously, I think many of you guys are probably doing something very different from your capstone than what Gina's doing. But this is why it's also good to see other projects like this. And what's kind of cool is too, is it's not that these are very different, in the way you'd want to approach them, finding someone's work, seeing what's there, just like Jeff was telling you, Gina, for your capstone, you don't have to do something that's completely novel that no one else has ever done. It's you can try to reproduce this work. And the important part is really I tell people for capstone, it's about, it's the thing that you tell people oh, tell me about a data science project you've done. And you can talk about that and be able to talk about the nuances of why were these decisions made? Why was this good? Why don't we think this was good? And those are the kind of things you want to bring, because that's kind of what you'll do mostly, trying to figure out on a problem, it's going to take a long time, even if you do really, really well on it. It's going to be iteration on top of it, and the kind of exploration, and kind of like thinking like, like what would be the better way to do this, or like what's going on here. So this is really great. So I don't know if anyone had initial reactions that they wanted to share on this, but I think this is a really cool one. I think that like you kind of hit there, Gina, right? It's the ATCG, right? It's like that's the thing that's going to really like matter a lot. Like in this part, it's going to identify. The binary classes definitely are going to be helpful features, or at least I assume they're going to be helpful features.
They will be.
In that. Yeah, and it kind of makes sense, like you were saying with random forest, is that basically like using like equivalent to an n-gram, right? Is really what's going on here. So anyone who's doing like NLP stuff, this is essentially what's going on here. And you can think of like A, C, G, N, and T are essentially words in a sentence. And basically this, like 3 different, letters, right? 3 different nucleotides.
Nucleotides, yeah.
Okay,
Very good.
I'm not a bio major, but I remember enough from like my like AP bio class in high school.
Yeah, there you go.
Yeah. But those could essentially be words, because in a sense, like they are individual. In this case, you only have like what, 5 different words or 4 different words. Like are nucleotides,
well,
that at least for this problem, like
for this problem, and the other thing is, by the way, so the 4-letter repeats they did them as permutations, and I'm looking at them going, wait a minute, they don't have repeats, and that's part of why I picked 3, because 3 corresponds to, as I mentioned, each 3 base pair corresponds to a nucleotide, to an amino acid, and then also start and stop codons are 3, generally tend to be 3 as well. And so what I ended up doing was doing, what do you call it, combinations as opposed to permutations for my 3-letter. So that gave me close, like a similar number of things as what they had, but that allowed me to have some repeats in here, as you can see.
right.
Yeah. So
Yeah,
so I don't know if it's so much that each letter is like a word, but
I was going to say, like, just for
kind
like
of,
when you think of
maybe.
n-grams, like, oh, that's essentially what's
Yeah.
going on, right? It's kind of like,
Okay.
oh, if you had word, 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 it's kind of like these words in a row. And so it's like these ones in a row. And so this can help, like a lot of ways you can talk about, like, just like a sentence, right? With NLP, it's like, oh, we can reduce the complexity of like a full sentence of this full, genetic sequence. We can reduce the complexity by doing something like n-grams or something along those lines. This, if you think of it in terms of NLP, that might kind of give you an idea of like other strategies that might not necessarily work well, but like could potentially do stuff. Like you can essentially say, like, what's the count? Like, how many times does A occur in this sequence? How long is this sequence, right? My guess is that, this might actually be a useful thing to say, hey, like, does that do just as well as like doing like a random forest on, 3 or 4 letter sequences? Because if it does, you're like, oh, we're hitting this baseline. We need something a little more powerful, which makes sense when you're saying recurrent neural networks, RNNs, right? The big thing about recurrent neural networks makes sense is that it's about a sequence, is that order matters. And so, and if you guys know anything basic about like DNA genetic sequence, it's like, oh yeah, there's something about the order at least to code what ends up being whatever makes this unique, right? And so,
and you
in
can
that
see
case,
here the counts, like, so for example, this, I think this one's interesting because I think it's a restriction enzyme or a primer. There's lots more data in here, but you can see, like, for the smaller ones, like AGCT, and that corresponds to something manipulable. And you can see this sequence has 39, this has 2, this has 3, 24, etc. So sorry to interrupt, but I wanted to talk about like what you said. So it's just counts here. It's not really talking, I mean, for this initial data manipulation, it's not talking about order at all, except for those that explicitly lay this out.
And I think that's probably where you'll probably reach, which is why you're like doing a few different strategies and they're more or less about the same. But probably to get like above that is probably to start thinking about like what happens when we consider, essentially structure of the actual like order of it. Because we're capturing a little bit by doing, segments, essentially like n-grams here. So one way you can do this, is recurrent neural networks. That's kind of like I think that seems like the most like natural if you think about like what we're looking at. It's like, oh yeah, like a sentence, recurrent neural networks, LSTMs, that kind of makes sense, sequence. There is a strategy that potentially could be used here, which is kind of interesting. Because when we're looking for like structure, right, we're talking about like order of sentence, but what other things do we use for structure other than like natural language processing? Like has like some kind of like structure beyond just like what's in it? And that'd be images. So images, right, like pixels that are close by each other matter more than just saying what the individual value of that one pixel is, right? So you can actually like, you could potentially form this in a way of a convolutional neural network, is that you can use that and structure it as if you're using like, you know, essentially your pixels are essentially the values of A, T, G, C, you know, and whatever else you're using. So that's one potential thing. I think this can be useful for sometimes variable length, what's it called? Variable length, like essentially arrays, right? Which is what you have here. Some sequences are going to be very large, some sequences are going to be very small, and that at least will help capture some of essentially the structure of it. And so you can kind of think, let's say something really simple, like you have like 9 letters, right? 9 nucleotides, and we put those into a 3x3 grid. And let's just pretend, just to make things simple for us to think about it, you have all of them in that size. You can say like, well, you're not going to have like, it literally doesn't matter like, oh, like let's say A, T, T, G, C, right? Or something like that. But A and G is like, well, does that really matter? It's like, well, not really, like not the way DNA works, but it might be enough to give like an idea of what that structure is, where we have essentially something that would encode for an amino acid and then another amino acid. Like maybe those two together being strung together might be something that's like, oh, this is like the identifier that kind of helps doing that. So that could be a way you could actually use like something like convolutional neural net, where you can use a filter where it's going to look at something like multiple, you know, a 2D convolutional neural net or convolutional layer could be used on that. So you can look at maybe like 4 at a time, and that could be enough structure. The hard part's going to be is like, well, what happens when you have a variable list, right? And that's where you could do padding, for example. So you could potentially center it in the middle. And so that way, you know, like you could have like, if you have, let's say up to like, I'm just going to make up something like, you know, 20 by 20 or something like that. But then you only have like 9 ATGs, right? In like this part, you would center maybe that 9 in the center of there. And then basically putting padding of like essentially like a null character, or like it would be like a 0 or like something to represent saying, oh, this is an empty space. And you could potentially do something like that for like a convolution. So there's like some kind of like, and that's the kind of idea that you want to bring a little bit, is it's not so much about like, one is like domain, like this right here, like you have on the screen, that's domain knowledge, right? It's like, oh, you know what sequences matter and stuff like that, and looking to see those matter, you know, like can we actually identify from here? And it looks like it ended up being like any more useful than what you had before, and it's probably because you just need that structure in there, right? And so once you like recognize like, oh, it's a structure issue, like it's about the sequence of like order and stuff like that opens up to say, okay, what other techniques do we know that can be used on this part? So you can talk about like what kind of models to use, and that's kind of like what you might want to start off with first. I like to use like random forest, like kind of like what you're what you saw here and what you're kind of doing on this part, because it's a very quick thing for us to do once we have like a set number of features. It's very great for tabular data. Random forest will not do well on like, you know, pictures, because it just doesn't understand structure. It's not meant to do that. But you kind of talked about like, you know, what models to use, you know, what would work well. And this is where I kind of tell people like, there's kind of like this little dirty secret in like machine learning, is that it doesn't matter actually as much as people think it is to what model specifically you choose to make good predictions. It turns out there's I can't remember the theorem. I always forget it too. But you can probably look this up. There's a theorem that basically says is that if given enough data and given enough time, all models converge to about the same accuracy. And so, and like that's actually like a mathematical thing. It's like, oh, like this is like a legit thing, and you can see this in some data too. So what usually the model is about is saying, what resources can you provide to train this? What kind of model does this produce? You know, is this a relatively small model? For example, if you can do sparse matrices with this model, that might be really beneficial, because you don't have to have a huge model at the very end of the day. So that's all the stuff. Yeah, so I think more thinking about like what the structure approach, if that makes sense. Like, and I think the tabular, like if you think maybe this in like terms of NLP, that might give you a few ideas of like different ways you can maybe like, you know, possibly like, you know, like think of like again, I'm not like someone who's like a biologist, so I can think like, oh, like, is there an equivalent to stop words that might be like really frequent, but like, occur almost all the time, where it's like maybe it's not really a great identifier. I don't know. But like you can maybe think of in those terms of like NLP that might be helpful, and thinking of different approaches you could take.
Cool. Well,
Yeah.
thank you so much. And thank you guys. I'm sorry, I'm gonna have to jump off now, but and it looks like I scared Michael Matthews Jr. away. He left. But yeah, so thanks guys for indulging me, and I look forward to actually watching the rest of the video to hear about like whatever questions you have and how you solve them. And congratulations to Jessica about her second blog, which I saw in the other video. She got another one on Medium. So maybe a question for another time. What's your secret? And I haven't put stuff on Medium yet. So
You
anyway.
should, if you're feeling it for it, you should go for it.
Okay.
But
Yeah.
yeah, thank you so much, Gina, for sharing. I really appreciate that, and hopefully you all found this informative too, and seeing something a little different. So
cool.
And
thank you,
Gina.
Thanks.
This is Jessica. Thank you.
Okay, let's see. Probably should unshare. I don't know if I need to. Oh, stop share. There we go. Okie doke. See you guys later. Bye.
Take care, Gina.
Bye.
All right. Yeah, thank you all for like doing that too. Like, and this is something like, if you guys have problems too, like, or like not even problems, just like, oh, I want to talk about my specific problem. Don't feel like you can't talk about that, because I mean, hopefully you guys found that informative too. And, you can see a little bit like when you come across problems that like, like how to use it. Like, it's not so much saying, oh, this is like a certain problem, so I need to throw this specific machine learning algorithm and do this. It's much more thinking about what is the underlying structure of this data, and how can I take advantage of that structure to basically do prediction or whatever else you need to do. And I know that's kind of tough too, especially as you're learning a whole bunch of different stuff to use. But hopefully that makes sense where like, like I think a big part of that, like what she was showing, like the different sequences, thinking in terms of NLP, thinking about like n-grams and like say, well, okay, this is kind of related. Thinking about it possibly like convolutional neural nets, like, okay, looking for structure, that can be really helpful. So.